So unfortunately for you, chapter 9, like chapter 8, is like 2 or 3 chapters of Bio 1 all condensed into one chapter. So it's really long. Well, I'm just going to try to, we'll break it into a bunch of parts. The first part is going to be DNA structure, which that's the reason we watched that movie, that short film, so that I can talk about the structure a little more quickly. And then replication, which is what Crick was speaking about there at the end in that old sort of grainy film, right? When he was talking about how you could separate the 2 strands and make a complementary copy, and it would be how genes are transmitted from one generation to the next. And that was a big deal. It doesn't seem like it to us now because you've known about DNA since you were like in 4th grade, but it is a big deal, right? So it's the basis for the vaccine that you all hopefully got shot in the arm, right? So they're making multiple copies of messenger RNA from this virus because we know how replication works, right? Because we're not to be too graphic, but we're bastardizing the enzymes that are involved in replication and using them to do something else. So we're going to do this first, and then how the DNA stores the code to make the proteins. So we'll have to talk about messenger RNA, right? Transcription. And then how that messenger RNA is translated into amino acids / proteins, right? How we build it into proteins. That one's called translation. Remember that one with the messenger RNA and the transfer RNA and the ribosome? Just nod your head, it'll make me feel better. So yes, of course we remember it. So we've kind of got the structure of DNA. What is the code? How is it? How do we hold it? How do we transmit it? And then how do we make proteins out of it? Remember, proteins do everything in cells. So we spend a lot of time making proteins if you're a cell. And then the 3rd and 4th section of the chapter, there's a section about regulation, about gene regulation, which some of you may have gotten in Bio 1. I don't talk about it in Bio 1 because I feel like it's enough if we can get through this. But we're going to talk a little bit about it in here. And then the last section is about mutations. So we kind of have these big 4 blocks to kind of master in chapter 9, which is why we're just going to have chapters 8, 9, and 6 on a test, right? You're just like, that sounds like a lot. Well, hopefully you're already working through chapter 8. And again, it should be a refresher from Bio 1 and from Chem 102. I did want to say before I forget that I went through yesterday and I gave everybody extensions who either didn't complete the Connect or had like 20% of it completed or 35% of it completed and then just kind of gave up. So if either of those are your situation, please go back in and keep plugging along, okay? It's a pain, it's boring, I know it's time consuming, but that test is over, I get it. But it is going to be whatever I said, 5% of your grade or whatever. And it's really like free extra credit that helps you get better grades on the test. So it's to your benefit if you do it. There is a light at the end of the tunnel. After chapter 12, I don't do any more Connect for about 3 weeks. And then there's a little bit at the end for the diseases. So there will be a break from it. So know that it's not going to be this slog for the whole semester. It's especially heavy at the beginning because there's a lot you need to remember. And I get it. I was a student once too. And we think you should remember everything that you've been taught before. The reality is you study something for a test and then a lot of times it just leaves you, especially if it's not important to you, right? Which, I get it. This is not. I understand. So do your Connect homework. Okay, so let's start here talking about structure and replication then real quickly, okay? So like I said, that's the main reason that I showed you that video. So we always say, right, 3 things about DNA, right? It's double-stranded. that's like, you know, a 7th grade way of saying 2 chains of nucleic acid. Remember, the nucleic acid is the chain, the nucleotides are the individual building blocks, right? So we say it's double-stranded. This is often where we mention that these strands are complementary, right? Meaning a purine always pairs with a pyrimidine. Short word, 2 rings, big picture. Long word, short picture, 1 ring. So it's always a 2-ring molecule pairing with a 1 nitrogen containing ring molecule. Does anybody remember which ones are the purines? Which ones have the 2 rings? I'm not hopeful. I'm just throwing that out there. Take a guess. It's 1 and 4. Not thymine. Adenine pairs with thymine. So thymine must be a pyrimidine. So you got, now it's down to 50-50. How many for G? For guanine. How many for cytosine? It's guanine. I vote both times so that you don't get swayed. Yes, so it's guanine is the other purine and cytosine is the other pyrimidine. Between A and T, we have 2 sets of hydrogen bonds. Between G and C, there are 3 most of the time. Why do you care? Because that makes GC bonds a little bit tighter. And if you have a run of DNA where there's lots of G's and C's, it makes it harder to pull it apart, harder to, sometimes it's an indicator of where a gene starts or where there's a transcription factor that binds, because this is a little more stable. But come to genetics, learn more about it. All right, so they're complementary. That comes to us, that was when they were talking about Chargaff. Remember that? They showed us a picture, right? Guy from Chicago, or no, guy from, I forget where he was from. He purified DNA from sperm cells of all different kinds of animals and then analyzed how much adenine, how much thymine, how much guanine, how much cytosine. And that's what he figured out and reported was the amount of A was a percent of A was approximately the same as the percent of T, not exactly, but close. And the amount of G, the percentage of G was close to the amount of C, right? We call it a double helix. And that doesn't mean much to us, but it means something if you're a biochemist, right? That's what they were talking about when they were talking about the 2 strands run in the opposite direction. And there are areas where there's a big, we call it the minor groove and the major groove, right? So the double helix part has to do with being anti-parallel. The strands run anti-parallel to each other, right? And we'll talk more about that in a second. I would just remind you that the nucleotide up here, sorry, is made of a nitrogenous base, a sugar, and we leave it vague on purpose, but in DNA, that sugar is deoxyribose, right? Because it's missing an oxygen and a phosphate. And the A, like we said last week, A is the same A that's in NADH and ATP, and G is the same G that's in GTP. As you progress, as you go along, if you go further on in biology or chemistry, what you'll find out is a lot of the time when we tell you in metabolism an ATP is produced, it's actually a GTP. But it just kind of depends on who's around, like what molecules nearby that the extra phosphate gets thrown on. They have the same amount of potential energy. We just call everything ATP in undergraduate because we want it to be easy for you, not have to remember all different kinds of molecules. Okay, double-stranded, complementary, anti-parallel, double helix. All right, let's talk about what anti-parallel really means. All right, that's what she saw in the X-ray crystallography. That's what they figured out from photograph 51 that Rosalind Franklin wasn't working on, because that was the B form of DNA. She had isolated and taken X-ray crystallography pictures of A form that was a little drier, meaning with a little less water, and it didn't form the same beautiful helix quite as easily. We call that the A form. There's also a Z form, but only a few oddballs study that. So what anti-parallel means is what's on the ends of a chain of DNA. We call them the 5' end and the 3' end. Yes, you've heard this before. Yes. Okay. 5' end has a phosphate on both the 5th and the 3rd carbon of the last deoxyribose. I'll show you a picture in a minute. I'm not going to draw that. All right. The 3' end of a chain of DNA still has a phosphate on the 5th carbon of the deoxyribose, the last deoxyribose. But it has a hydroxyl group, OH, on the 3rd carbon of this same deoxyribose. And that's enough chemical difference. I have a phosphate with a big negative charge and some oxygens on it, or I have an OH group, which is a lot smaller, right? That's enough of the chemical difference to tell, that's going to regulate how all the enzymes for replication, transcription, and translation, they all work the same way, because all of them recognize a free 5' phosphate. So it's kind of important, right? Because it gives an orientation to how the code is read, how the code is stored, how the code gets made into proteins. What's the order of the amino acids in the protein? Yeah, so like I said, I'm not going to draw that picture. I'm just going to show you that picture. Maybe. Where would that be? Okay, that's annoying. Yeah, move it. Thank you. I need that. I need help doing that stuff. I need like a room arranger. It's annoying, isn't it, when it's not where you want it? I totally understand. Try this one. Glasses would be good. What chapter is this? 9, right? There we go. PowerPoints for chapter 9. Let's take a look at those. Okay, can you see that? Good. Let's look at this picture. It's my favorite one. And if I share screens, can you still see that? You can still see that, right? Okay, good. Yeah, that works. Make it really big. Whoa. Slideshow big. There we go. So if we just take a quick look at this, which is whatever it is, figure, if you're at home, figure 9.4. The reason I put this up here is to show all those things that I just talked about, as far as the structure. So we have the complementary base pairing, A's with T's, G's with C's, right? We also have the 5' end shown here, where this is the sugar, the D with the yellow, and we have the phosphate on the 5th carbon, phosphate on the 3rd carbon. So this would be a 5' end. And the other end of that chain has a 3' hydroxyl. We say the chains are anti-parallel because the complementary chain runs in the opposite direction. See the arrow going up? So the complementary chain has to have a 5' end across from the 3' end and a 3' end across from this 5' end. Convention says when you draw a picture of DNA like this vertically, you always put the 5' chain on the left. If you see a piece of DNA horizontally, hot dog way, 5' chain's always on the top, right? That's the 5' direction. And that's just kind of convention. So you can kind of remember that for later. Here's the double helix, what it looks like. Here's the minor groove. Here's the major groove. Can you see that those are different distances, right? That was also shown in Rosalind Franklin's X-ray crystallography picture. And usually, sometimes, not always, but sometimes it helps. I like to do it this way because that's the way that Crick did it when he was explaining it to Watson. He had to just regular number 2 pencils, right? But he said they were building the molecule like this, 5', 5', if the pencil points are 5'. And when Crick saw the photograph 51, he was like, no, we're supposed to be building it like this, right? And just that chemistry change of putting the 3' end across from a 5' end, that gave them the structure. That gave them what the helix that matched the X-ray crystallography. Okay, so if that helps, if not, whatever. Okay, now we have to talk about replication. How does this molecule get copied? This all stuff you've heard before, yeah? Yes. Right. All right, so now let's talk about replication, which hopefully will add a little bit to what you know, or if not, hopefully you'll just learn some new stuff. All right, so replication. Right, so to copy, to replicate. And when we replicate the DNA, we have to copy all of it. So we're going to make a copy of all the DNA in the cell. If we're in a bacteria, that's a lot less DNA than if we're in a eukaryote, right? Because bacteria have that one circular chromosome that has maybe 4,000 to 6,000 genes on it. And you have 23 chromosomes with 25,000 genes on them, between 25,000 and 30 genes on them. So you have a lot more material that needs to be copied. So we'll just look at a little piece at a time. In order to do this copying, we call the way that replication happens, we refer to it as semi-conservative. Like they pictured in that short video clip, the DNA is going to unwind and unzip, and we're going to use each of the parent strands to make a complementary new strand. Right? So I'm going to draw just real quickly. I'm going to draw in my black marker here. Right? The lines being the base pairs, A pairs with T, C with G, this being a 5' end to a 3' end. This being 3' to 5', I should have made that a little better. Should have ended it here. Right. 3'. No, anyway, I can't do that. Sorry. I know it doesn't look like it's right, but when this unwinds and unzips, so I'll put the 5' one over this way. Yeah, I need some spatial drawing help. That. Right, so this one's my 5' to 3' strand, this guy, and this one is my 3 to 5. Right. Each of them is going to get a complementary. Let's make it orange so we can see it. Right. So where there was an A, the new strand is going to have a T. Where there was a G, the new strand is going to have a C. Got a little thicker, so you can see it. That's why it's semi-conservative, okay? Because half partially is conserved. So each of the new cells, this is, if you're in a eukaryote, this is mitosis, or if you're in a bacteria and this is binary fission, right? Each of the new cells is going to get an exact replica of the original, unless there's been a mutation, right? So that one guy with the beard was talking about the mutability, right, as the basis for evolution, as well as the conservative nature of pretty much preserving the same sequence over and over again. Okay, so this is all regulated by a set of enzymes. Really long names. There's a table in your book. It's in purple, I believe. It's on this page 234 that lists the major enzymes and their functions. I would take a look at that. I'm going to write words and draw a picture, okay, to sort of go through the process as best I can. So erase this. I just feel like we're going to do that. Okay, this marker is getting removed into the do not use pile. All right, get rid of this and write some bunch of words. All right, let's start, like, we'll number them as steps in a linear way. Okay, first thing that has to happen is the DNA has to be unwound. So we have to undo the helix part, we have to make it straight, and then we have to break all the hydrogen bonds. We have to open up those two strands. So we say unwound and unzipped. The unzipping is the break the hydrogen bond part. All right, and remember, of course you remember this from bio. Individual hydrogen bonds are weak and form and reform, but when you have hundreds and thousands of them together, like in water, right, they give you new properties like surface tension, and takes a high specific heat to break water molecules to make them change from liquid to gas. Same with DNA. If I just have one hydrogen bond, no problem to break. If I have two between each A and T and three between each G and C, that gives me a lot of hydrogen bonds to break, and it's not going to happen naturally. In order to do this in the laboratory outside of an organism, you need to heat the DNA to 95 degrees Celsius, almost to boiling, to get those hydrogen bonds to break. Okay, and you know you can't do that if there's proteins, right? Because proteins would denature way before that. So this is a cool thing, that there are enzymes that can just break those hydrogen bonds. The unwinding is important because if you don't unwind it and you try to just unwind a little piece of it, you're going to make so much tension in the parts that are not unwound, we call it supercoiling, that it's going to break. And that would be no good, right? We need the DNA in this order so we can make these proteins in this order. And if pieces of it are breaking off and just floating out into the cytoplasm or reattaching in the wrong direction, that's not good for the organism. Okay, so the unwinding, we say, is done by an enzyme called DNA gyrase, which is kind of easy to remember, right? Because it gyrates, spins. And the unzipping, helicase. And these two enzymes are going to be kind of out in front of the main replication machinery. Your book is going to name DNA gyrase, but they're going to tell you that it's in a group of enzymes that are called topoisomerases. And basically that means they can change the topography, right, of isomer, chemistry word, right, from one form to another using the same molecule, just like the D form of glucose versus the L form of glucose. Right. So there's a whole bunch of these. There's a whole category of topoisomerases, like 8 of them or 12 of them. I just need you to be able to link that word with gyrase. Okay. So in my picture here, I'm going to draw. What I had. Three sort of stages of this. One with the DNA barely open and just getting started at replication. Another one where the DNA is about halfway finished being copied. And yes, I'm drawing this as a linear thing because it's probably a little bit easier for us to understand. If I draw here, can you still see or no? No. No. You specifically.
No, but it's fine.
You sure?
Yeah.
How about if I make it very close to the other one? And if I put way out here, right? You can see it. All right. So if I draw the lines, right, to make the base pairs, and I would encourage you to do this too, either now or later to practice to help you remember. 5 prime. So hopefully this will be a 3 prime end, and then that makes this a 5 prime end. 1, 2, 3, 4, 5, 6, 7, 8. Let's just put 8 bases there. And same, I'm going to put 5 prime, 3 prime. And I'm going to put bases along here. The lines that I drew, remember those are the phosphates and the sugar. The bases actually attach to the sugar, not the phosphate. And I'll try to draw these a little more neatly, but then the ones on the bottom, probably not as much. Yeah, you get the idea. I'm tired now. All right. Okay, so first thing that has to happen, we need helicase and gyrase to unwind and unzip. And I'm going to look for my other orange because hopefully it won't make a mess on the board. So helicase and gyrase need to be in front of where we call this the replication fork. They need to be upstream of that, right? Before, so it's already happened. So I'm going to put helicase right here. I'm going to draw it kind of as a rectangle. And gyrase is actually a little bit further upstream. And gyrase, remember, is relieving that supercoiling. So if you've ever made, make those little friendship bracelets where you weave the colors together for your friends. No, yes, you did that once when you were small, right? And it says, you know, you need 3 strands of blue and 3 strands of orange of the 6 strands of the embroidery floss, right? So you cut a big piece of floss and you try to pull the strings apart, or maybe you've ever tried to pull rope apart, right? Something that's wound and twisted, and you pull, and at first it's really easy, and then it wads up into a knot. That's supercoiling, right? And just like it can break your thread or make that part of it unusable, the same thing would happen to DNA. So what these topoisomerases do is they either make a single nick in one of the strands, or some of them actually nick both strands, and they let the DNA do this, unwind, and then they seal the gap so that it's not supercoiled as a consequence of this unwinding up here. Okay, so we've got helicase and gyrase checked. Unwind, unzip. Next thing, now we're going to put an enzyme on there, and the enzyme's name is primase. And this is difficult because it's a primase, ASE, it's an enzyme, right? That's what should make it purple. What does it do? It makes a primer, and the primer is an RNA primer. And this, you're like, okay. So remember, RNA has ribose in it, not deoxyribose. RNA is usually single-stranded. And a primer, what's that? It's like a choke switch on your lawnmower, right? A primer is a short piece of complementary nucleic acid. So in this case, it's a short piece of complementary RNA, right? I'll put RNA in the parentheses here. I like to make primase pink. So let's see if my pink has anything left in it. So primase is going to sit down here, and I'm going to put it a little further down because I'm going to have it already have made the primer. It's going toward the replication fork. So the pink ball is primase. It's headed toward the replication fork. It is making DNA complementary and anti-parallel 5' to 3'. Ooh, that's a 3' end. Perfect. So primase is recognizing the free 3' hydroxyl here, and it's going, oh, I'm going to sit down there and make a primer. That's what it does. Short. How short is short? 20-ish. Usually we say between 11 and 25 bases for a primer. I'm giving you a range there because it does differ for different organisms, right? And a lot of times use primers in molecular genetics to do other things, right? So it can be longer or shorter than that. Okay, so we've got primase. Now we have a primase, now we have a primer, now DNA polymerase. And we're going to talk about DNA polymerase 3, I believe. Right, because your book's going to split them into 3 and 1. Yeah, okay. DNA polymerase 3, which is often abbreviated like this, DNA pol 3, right? Because it takes too long to write polymerase. Bacteria have 5 polymerases. We're going to learn about 2. You have 8 polymerases. We're going to learn the same, right? We're just going to learn about a few of them. So polymerase 3, DNA polymerase. So what is it? ASE? It's an enzyme. What does it do? Adds DNA, makes DNA polymers, right? That's what it does. DNA polymerase 3, we need to note, recognizes the 3' hydroxyl. Well, let's not say that. Let's just say recognizes the primer. The RNA primer, RNA DNA primer combo, and begins adding DNA nucleotides 5' to 3' direction. Cool fun fact. Does that between 800 and 1,000 bases per second. What? And the other cool thing that DNA polymerase 3 does is it proofreads. So it's a gigantic enzyme. And if you look at the physical enzyme itself, big, it has some subunits. The front end of the enzyme, if you want to think of it that way, is adding the nucleotides. The back end of the enzyme is checking. Okay, that was an A across from a T, a G across from a C. And if there's a mistake, clip, cuts it out and puts the right one in. On average, it's so good at adding bases at that speed and proofreading itself for mistakes. On average, it only leaves one mistake, one mutation in 1,000,000 bases. 1, which translates to about 1 mutation in 1,000 genes. So E. coli has 4,000 genes, so that's like 4 mutations every time the bacteria copies itself. 1 in a million. I mean, that's such, we call that high fidelity, a high fidelity enzyme because it's very accurate, right? Okay, let's make polymerase, and I will make it purple because it's a giant protein. I don't have room to draw polymerase here, so I'm going to draw it like right here, like that. DNA polymerase 3, right, heading this direction. The primer is technically still there. Another enzyme is going to come along and digest that. I'm just shortening it up a little bit. So DNA polymerase would have sat down here at this free 3' hydroxyl, the primer left, primase left, and it would start synthesizing DNA this direction. A's with T's, C's with G's, blah, blah, blah. 5' to 3', right, toward the replication port. And in my last picture, polymerase 1 is what's going to get rid of this primer and fill it in with DNA. And so in my last picture, it would look like this. It would all be DNA, right? And I would put DNA polymerase 3. Now I would put it here because it's sliding along the chain that's opened up. Helicase and gyrase are also sliding along. Actually, helicase kind of looks like a donut. I shouldn't have drawn it a rectangle, right? It's always kind of just here at the replication fork. And gyrase is always somewhere upstream of that, relieving supercoiling. So I'm just going to label those again. Gyrase, gyrase, helicase, helicase. Lovely. Just going to keep going. Let's mention, let's put in a mention for DNA polymerase 1. Right, removing the primer, closing the gaps, and repairing any mismatches that DNA polymerase did not fix as it was going by. So I'm going to put DNA polymerase 1 here. I'm also going to kind of make it roundish. All right. Oh my gosh, I forgot something. So busy thinking about the polymerases. DNA polymerase 1. Removes primer. Remember, that's RNA. And replaces it with DNA. I only thought of it when I did this marker because it's like almost a brown color, and I always use brown for single-stranded binding proteins. I have to change my plan here. I don't know, it's like a mind blow. All right, so after the unwinding and the unzipping, there's something I forgot. 1A, SSBP, standing for single-stranded binding proteins. Single-stranded binding proteins. They're usually in pairs, and they bind here at the replication fork. So I'm adding them in here in green. And their job as the DNA is further unwound and unzipped, there's more pairs of them jump on. Right? Their job is to hold the hydrogen bonds apart after helicase breaks the hydrogen bonds apart, because hydrogen bonds form spontaneously. So if we break them like a millisecond later, they're just going to go whoop and wind back up. So when helicase breaks the two strands apart, the single-stranded binding proteins jump on and keep the two single strands of DNA open at the replication fork. So they're always kind of close to that replication fork. And they also are kind of like donut shaped because they kind of slide along. Can't believe I forgot that. Okay, so that's good. Do you notice something about the picture, right? We've only copied the top strand, right? Because this is the easy one. And because this strand was the 3' strand, so it's very easy for primase to find that free 3' hydroxyl, jump on, make the primer, and get started. But we have this whole other strand that we have to copy. And you see how there's a little bit of an anti-parallel problem here, right? I cannot put primase here and make another strand 5' to 5, 5' to 3', because then I would not be anti-parallel. Right? So I'm going to say that again, because I get that for some of you, this might be the first couple times you're hearing this. Primase cannot sit here and go this way because I would make a non-anti-parallel, I would make a parallel strand instead of an anti-parallel strand. I can't have 5', 5'. I got to have a 3' base here, a 3' hydroxyl. So the other strand, we call that the lagging strand, because it seems to be made a little bit more slowly. And I'm going to erase these steps and put the lagging strand steps on there, but probably with fewer words, because some of it repeats. Okay. Plus, it's good for you to write everything again, because it helps you remember it. The more times you write it, the more likely you are to remember it. So I'm going to label this lagging strand. Right? And for your reference, the bottom one in our picture, or the 5' to 3' strand, to help you remember. Okay. So how are we going to replicate that one? Well, still involved is primase. And there's something I didn't tell you about primase. Primase, remember, it makes an RNA primer. So in reality, primase really is a DNA dependent RNA polymerase. So it's really an RNA polymerase, but we don't want to tell you that right now. And what RNA polymerases can do that DNA polymerase cannot is RNA polymerases like primase, they can see, recognize the 3' side of a nucleotide. So let me just try to help you with that a little bit, right? So you've seen railroad tracks in your life, right? So think of the rails on those railroad tracks, like the phosphates, right? And the sugars. And wherever there's a railroad tie going through, that's a base pair, right? And you maybe walked on a railroad track before or looked at it and know that there's a gap between each set of ties, of railroad ties, right? So think of this primase as sliding down the rail. And when it's standing in between two bases, two railroad ties, it literally can look right and left and say, oh, that's the 3' side of this base and the 5' side of this base. I'm going to start here and jump in there and start synthesizing a complementary copy. Okay. Even though it's very close to where the break is at the replication fork. So for the lagging strand, what happens is primase jumps on at the replication fork and makes a little primer this way, 5' to 3'. Going away from the replication fork. Right? So it's still going to be sitting here in this picture, but DNA polymerase is going to jump onto this. I should have made it so long. It's going to jump on here and make complementary copy of DNA. And so then we're going to need another primer at the replication fork again to provide that free 3' hydroxyl for DNA polymerase. And I'll just draw DNA polymerase once here, going this direction, 5' to 3', like that. Remember, it's DNA polymerase 3. It's going to use this free 3' hydroxyl to begin adding a 5' base. So let's write some words to go with it. Primase makes RNA, it can recognize the 3' side of a nucleotide, and on the lagging strand, it is going to be working at the replication fork. I think of it out or away from the replication fork. So it's going to make a primer, 5' to 3' short. Then DNA polymerase 3 is going to recognize the primer and begin adding nucleotides, complementary, anti-parallel. It's only one R in parallel, and those 1, 2, 2. Right? Away, again, away from the replication fork. This piece that DNA polymerase 3 just made that's bound on the edges by primers, that piece has a special name, right? Anybody remember what it's called? Ends with an O. Yeah, Okazaki fragment. And I'm going to call it Okazaki fragment A. Still, well, that's just. They're in the neighborhood of about 200 plus nucleotides. So from the replication fork till DNA polymerase runs into the next primer, if it's still sitting there, that is called an Okazaki fragment. Okazaki fragment. I like to take this moment to point out that that's really significant because when DNA replication was being sorted out amongst the scientists, all of the enzymes had people's names because everyone thought they were so important, right? After it was all sorted out, they're like, no, we're going to rename these enzymes by their functions so people can understand what the hell's going on in this and not trying to remember 10 people's names, right? That's the only thing. It's like the Golgi body. That's the only thing that retains someone's name because he's the guy that figured out these things existed and how the opposite strand was replicated. It was that big of a deal. They were stumped for so long that when someone figured it out, we're like, okay, that's going to stay an Okazaki fragment. We're not changing the name of that. All right. So it's really important. It's like a big deal. Well, I think it's a big deal. So then still DNA polymerase 1 is going to come along, digest, right, the RNA DNA hybrids, fill them in with DNA. Okay, lovely. So I'm going to get rid of this primer here and fill it in with DNA. No, let's just leave that. Okay, so on this bottom strand here, I'm going to redraw. I'm going to put another primer here, 5' to 3'. I'm going to put DNA polymerase, right? Let's say about to there. It's just finishing up. DNA polymerase 3. That complementary, blah, 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 right? Doesn't show up very well, but I still like it. So this Okazaki fragment that's here that has now, the primer has been digested away and we have it left, this Okazaki fragment A, this one, right? This is the new one that just got made. Let's call that one B. Let's make those green. So this is fragment A, this is fragment B, right? Or fragment 2, if you want to think of it that way. The one thing that DNA polymerase 1 and DNA polymerase 3, the one thing they can't do is this. They can't join a 3' nucleotide to a 5' nucleotide. If they could do that, we wouldn't need primase, right? So we need one more enzyme on our chain here. Number 3, I'm going to put DNA ligase. DNA ligase's job is to join the Okazaki fragments together, right? I like to think of it as like stitching them together. So right here, I'm going to draw DNA ligase. I like to draw it as kind of a blue diamond, but it's not working out very well. DNA ligase. To ligate, ligature, to join, right? It joins those Okazaki fragments together. It's the last chemical bond that's made. So it kind of jumps. So it's here. And then as soon as DNA polymerase, as soon as we get more unwinding and unzipping and primer and another Okazaki fragment 3, ligase jumps from here to there to stitch the next ones and keeps going. All right. Take your pictures if you want to. I would highly suggest that you practice this on a big blank piece of paper or on your tablet or whatever you do, because the more times that you practice it and say it, the more times you're going to remember it, right? Oh, 9:16, perfect. All right. I'm going to end the recording real fast while you're doing your photographing. I will